Hi, I'm Lea Richardson, and today I'll be talking to you about some preliminary data from my postdoctoral work in the Yoder lab at California State University, Northridge, where I'm identifying sources of local adaptation in Joshua trees using genomic data. And so let me introduce you to the Joshua tree. You might already recognize it because it is quite a charismatic species that's pretty well known. It's a large tree that is native to the Mojave Desert. Obviously, there's Joshua Tree National Park in California, but the range of Joshua trees extends northern into the California desert up through the edges of Nevada and Utah and even to Arizona. And Joshua trees are pretty important in the habitats where they live because they're typically the largest plant, and so they provide food and shelter to many other organisms. There are two species of Joshua trees. There's Yucca brevifolia in the western half of the range, and then there's Yucca yageriana in the eastern part of the range. And you can see the western Joshua tree is a little bit more tree-like. Its branches come out of the base about 1 meter off of the ground, and the leaves are a bit longer. Whereas the eastern Joshua tree is more bush-like with multiple branches coming out near the base of the center trunk and slightly shorter leaves. And here's the range that I was talking about. Here you can see the lighter green areas that are shaded in are Joshua tree habitat, and the icons of the Joshua trees indicate the two different species. So you can see on the west, you have the western Joshua tree, the more tree-like one, and in the east, you have the bush-like tree. And on this plot, you'll also see, or on this slide, I should say, you also see these two moths depicted, and that's because Joshua trees have these tight obligate mutualisms with these two moth species, and it's a paired relationship. So this has been really interesting, as you can imagine, to evolutionary biologists for quite some time. And so this is another reason why Joshua trees are kind of a classical example when looking at coevolution. In this case, these two moth species, they rely on the Joshua trees, and in turn, the Joshua trees rely on the moths for pollination. So just to show you what I mean, here's a Joshua tree in fluorescence. And as a side note, Joshua trees don't flower every single year, but this year was a masting year for Joshua trees. So if you happen to find yourself in the Mojave Desert, you might still see remnants of the fruits from this year's flowering. And here you can see one of the moths. And so in the process of pollinating, the moths will also lay their eggs. And so that's what I mean by this kind of tight relationship. From the moth's perspective, they are feeling selection pressure to deposit their eggs at a distance that's close enough to the developing Joshua tree seeds that when the larvae hatch, they'll be able to access the food source. And from the tree's perspective, they obviously rely on the pollination services, but they also are evolving with the pressure of these moth larvae eating their seeds. And so we expect that these biotic relationships, this pollination relationship, is important in terms of driving evolution between these two Joshua tree species. But if we look at the overall range map, we can see that Joshua trees actually occupy quite a range of climatic conditions. Although these are all desert areas, there are quite a lot of differences in terms of precipitation, seasonal timing, for instance, in Arizona, where there are some more monsoon type summer precipitation. You also have trees on kind of the eastern slopes of the Sierra Nevada, and those are experiencing quite a bit different conditions than, let's say, a tree in Death Valley. And so these different climate conditions might also be exerting selective pressures on Joshua trees, leading to local adaptation. And these types of local adaptations might be important, especially from a conservation and management perspective. And so one of the big questions in the broader Joshua Tree Genome Project is looking at which sources of selection matter most to these Joshua trees, where we expect there to be a big effect of these biotic relationships, but also there could be these climatic factors driving Joshua tree evolution. And so for the work I'll be talking about today, we're specifically interested in looking at these associations between some of these abiotic potential selective pressures in terms of climate variables and how that might relate to local adaptation. And looping this back into the big picture, this is really important, especially for land managers and people trying to conserve Joshua trees, especially when you consider that Joshua Tree National Park is in the southwest range of the Joshua Tree range. And so trees there are projected to potentially not survive in most of the park within 50 years. So, you can imagine that land managers there are really interested in finding genetic material from source populations that will be well adapted to, proposed climate refugia within Joshua Tree Park if they're trying to kind of coordinate conservation efforts that will actually have a big impact. And so understanding this local adaptation is obviously of interest to a lot of different stakeholders. And so the Yoder Lab was able to get funding from Revive and Restore to conduct tissue sampling of a sample of 300 trees spread across the climatic variation experience in the Joshua Tree range so that we could run some whole genome resequencing. And so map is showing all these points are showing where the trees were sampled from. And I want at this point to just say that all these people pictured here did this genomic inventorying, driving around, collecting the samples, and then all of the lab work with library prep and everything for the sequencing, because all this happened actually before I even started my postdoc. So I was just handed the raw data set. And so I rely on all of these people and appreciate all the work they've done to get this going. So let's look at some of the climate variation we capture with the samples that we have. These are 11 plots that we don't need to focus on too much on the details, but they're all climate factors that have been associated in other studies with the presence or absence of Joshua trees. Things like mean annual temperatures or precipitation, seasonality of these factors, these temperatures and precipitation, maximum temperatures and ranges. And what you can see is all of these histograms are basically what we see colored by type. So the tan is the hybrid zone, the pinkish color is the eastern Joshua tree, and the darker maroon is the western. And then the vertical lines are just showing the means. So you can see that there's actually quite a separation in means of these different climatic variables in the different Joshua tree types. And so what we ended up with in the end was 298 trees that were sequenced using 5 lanes of Illumina NovaSeq. Then we aligned those sequences with Bowtie 2. We ended up with about 2.5x coverage, but upwards of 10x in some samples, and a mean map quality that was somewhat variable, but generally upwards of 60% of the bases were covered. So this is a low coverage whole genome sequencing data set, which means that we don't want to bias downstream analyses with our low coverage. And so we're going to be using angst, which allows us to calculate things like genotype probabilities and genotype likelihoods and use those in the downstream analyses to avoid the biases we might have if we were to hard call genotypes with such a low coverage data set. So far we've tried many filtering approaches to get a good quality data set of SNPs and genotype likelihoods. And so far we've identified over 16 million SNPs across the genome. And we've taken that information and run it through admixture using NGS admix. And so these plots are basically the result of, asking admixture to separate these trees into 2 groups, and we can see that they basically separate by color into the 2 different species. So this is exactly as we would expect given prior data on this species. And then the sliver that's on the left of the screen is from the hybrid zone, and you can see that there's a fair amount of admixture there as we would expect. We were also able to run our genotype likelihoods through PC angst in order to get this PCA plot. And so if you're not familiar with these, PCAs are basically showing you the differences between the points. So here each point is a tree. And so when they're close together, that means they're more similar to one another. And when they're farther apart, they're more different. So what we can see is that the 2 different species are more similar to themselves and a little bit more different from each other based on their spacing across these 2 PC axes, and that the hybrid trees kind of span that middle distance, which goes along with what we would expect. In PC Angst, you can also ask it to do an admixture analysis and choose the best K, which we did. And in that, it shows a K equals 4. So what that means is PC Angst was detecting these kind of 4 different distinct groupings, which when you plot them out on a map, nicely separate not only by east and west, but also by north and south. So we might be seeing kind of some evidence for these subpopulations. And in the future, we're going to run kind of a more rigorous NGS admix where we'll do multiple runs and choose the best K. And so that'll be really interesting to see what we get when we're able to do that. We also ran some preliminary diversity statistics on the whole data set, and we were able to use the sliding windows in Angst to calculate pairwise theta, which looks at the average number of pairwise differences between sequences. We were also able to calculate Watterson's theta, which looks at the number of segregating sites. And then with those 2 measures, we could calculate Tajima's D, which looks at the departure from neutral evolution by comparing the difference between the segregating sites and the average number of pairwise differences. So basically taking those bottom 2 metrics and looking at them in relation to one another. And so what we can see is points which are SNPs in the genome that are kind of outliers. So ones that are sticking out on the bottom or sticking out on the top are potential places where we might be seeing signs of selection. So balancing selection if they're outliers on the top, or positive selection if they're outliers on the bottom. So next steps are to obviously proceed with the genotype association analyses and see what those climate variables look to be related to, do more specific diversity statistics both within and among populations, and then we will move ahead and try to link all of those results back to the original kind of bigger questions about what types of local adaptation we're seeing in Joshua trees, what regions of the genome are involved in that, and use that to help guide restoration and management goals, which are increasingly important, not only because of climate change, but because these trees are facing, other kind of correlated problems like increased fire risk, as you can see in this picture where you have a bunch of burned trees. So, you can imagine managers are trying to find source material to plant out after something like a fire, and they want to be making informed decisions. And that's where this research really comes into play. And hopefully by next year, I'll be able to report back to you about the results of this study. And so with that, I'll take any questions. Feel free to email me. And yeah, thanks again to everybody that helped with all of this work.